So in this module, we're going to be looking at how you can view gene expression across multiple datasets. And in order to do this, we're going to use a function called the MegaSampler. So on the first page, in the first panel, you'll be able to select across datasets instead of single datasets. And you'll see here that MegaSampler and viewing a gene in more than one dataset is selected. There are other options that one can select. However, we're going to start with the view a gene in more than one dataset. So select that and click Next. So in the next step, we are presented with an option to select the type of data we would like to look at. And what this means is that you're going to select which platform the data has been produced on and the type of normalization or data handling that has occurred. And the importance for this being that if you are going to compare gene expression across multiple data sets, you want to make sure that the data that you're looking at has been processed in the exact same way, which makes it possible for the best comparison across datasets. That is not to say that this isn't a slightly biased approach, because we have not done any further handling of the data to normalize across datasets in this case. So we've simply made the assumption that if it's run on the same platform with the same type of data processing, that you should be able to at least get an idea of the gene expression comparison across datasets. So for this example, we're going to be using the U133P2 Affymetrix array platform with MOS5 handling, but you can see that there are multiple other datasets or dataset types that are possible to choose. So we're going to select this one and click Next. So in this panel, you are then presented with all of the datasets that have been run on this Affymetrix platform with the MOS5 handling. And you can see that the datasets have then been split up into different subheadings, including cell line, disease, experimental data, or tumor data, for instance. So for this example, I'm going to look at gene expression across multiple tumor types within the tumor datasets. So as you can see, there are quite a number of different tumor datasets that are available to compare. And for this example, we're going to just select a few different types of tumors. So we will select one breast tumor array containing 204 tumors. So we simply click there. Then we scroll down and we can select another array containing 290 colon samples. And then for this example, we will also select a neuroblastoma dataset simply for the ease of comparing it to certain gene expression profiles that we are familiar with from the previous modules. So we scroll down and we'll make use of, again, the Versteeg 88 tumor dataset that we've used multiple times. So now we've selected 3 different tumor types in 3 different datasets, and we're again going to look at MYC-N as the example gene in this case, and click Next. So as we have previously explained in the View a Gene module, you are now presented with multiple different probes that are all designed for MYC-N, and these are specific to this array platform that we're looking at, but we're going to select the one with the highest average expression. And then down below, you'll see that there are a number of options that you can set prior to viewing this gene. And here you can see that the data sets will be presented in a certain order. So we'll leave that as breast, colon, then neuroblastoma for now. And you can also make presets, for instance, on how you want to split them up according to annotations and so on and so forth. But we'll get on to that in the next section. So we'll leave all those as they are. However, one thing I am going to change here is I'm going to make them different colors strictly so that we can better visualize them in the next section. So you can simply click there. You can also adjust the colors if you know a specific color that you're interested in. Then you can type in that information here, for instance, and you can generate the colors yourself. And click Submit. So here you see that we are now presented with the data of MYC-N expression, the log2 of MYC-N expression across the multiple data sets. And not surprisingly, you can see that N-MYC expression seems to be highest in neuroblastoma, which is a tumor type that we know is associated with MYC-N amplification, so that's not so strange. As I mentioned before, you can split these data sets up into subcategories based on the sample annotations that are available within the data sets. So if we want to, for instance, split up neuroblastoma specifically into those that are amplified versus non-amplified to see how each of those groups compare to other tumor types, we can do that by simply scrolling down to the adjustable settings section. And under neuroblastoma, we're going to load the annotation, and then we're going to select MIC-N amplification. What we can also do here is that at the same time, we want to move neuroblastoma from being in the third place on the graph to being in the first place on the graph. And we simply do that by adjusting the numbers here. So I'm going to move that down to first place, and I'm going to switch it with breast and put breast cancers in the third place. So now everything else I'm going to leave as is, and we're going to redraw. And here you can see that now we have not only shifted the neuroblastoma datasets to the starting two positions on the graph, but we've also split it up into both the non-amplified tumors and the MIC-N amplified tumors here on the right. And as you can see that, yes, indeed, the MIC-N amplified tumors do have a higher expression of MIC-N than the non-amplified tumors. The standard non-amplified neuroblastoma tumors still do have a higher MIC-N expression on average than the colon cancer and breast cancer data sets that we are looking at here. So at the moment, we are looking at box dot plots. However, there are different options for different types of plots, just as in the regular view a gene section. So currently we're looking at box dot plots. However, we can, for instance, change that to looking specifically at a standard box plot. So you simply select that graph type. Again, we can change the colors as we wish. So we can make that one pink instead of red. And now we will click redraw. And you will see that you're now presented with a different type of plot. And again, the statistics or an ANOVA test comparing the differences across groups is presented above the graph. So now that we've looked at MIC-N expression, we can of course change the gene that we're looking at. So if you scroll down below the graph, here you can see find new gene, and we'll enter then NCAM1, which is a neural marker, simply to display that you can detect these differences across the different data sets. So we click search gene, and here you see that again we're presented with the different probes for NCAM1. We'll leave everything as is, just to recreate the same setup that we had for MIC-N, but for NCAM1. And here again, you see that there are quite large differences or higher expression of NCAM1 in neuroblastoma compared to the other tumor types that we have been looking at. So just to give you a couple more examples of how you can customize the appearance of these graphs, we're going to scroll down, and now instead of adapting by track, we're going to change this to adapting by groups. And just to give a broader view of how many groups we're looking at, we're going to change in the neuroblastoma dataset from looking at MIC-N amplification to looking at INSS stage, and we're then going to click redraw. So as you can see now, instead of just having MIC-N amplified versus non-amplified tumors within the neuroblastoma dataset, it's now split it up across the different stages of the disease, and those are now all viewed in the same color as we had labeled before. However, now what you can see is that you are able to adjust the colors of the individual tracks. So for instance, we can adjust stage 4 or the high-risk disease to red, and we can adjust stage 1 to green, and those in between we can make yellow. Just as an example, and then we can click redraw. And now you can see that we've changed the colors of the individual subgroups within the neuroblastoma dataset to match those that we wish to view. Furthermore, if we, for instance, only want to look at comparing the low stage versus the high stage, we can also go down below to the adjustable settings, and here you can simply turn off the stages that you do not, or turn off the tracks that you do not want to view. So now we're going to turn off stage 2, stage 3, and stage 4S, and we can now redraw this graph. And now we're only looking at stage 1 and stage 4 within the neuroblastoma dataset. And one additional note is that you can see here that these tumor datasets have all been labeled with a T in the beginning of them, but perhaps you would like to rename the datasets or label the axes in a different way. And you can see down here under adjustable settings again that These titles are indeed adjustable, so you can label these however you would like. So colon and then neuroblastoma. And we can simply click redraw. And now the axes have been changed to represent what you would like to display. So with the number of customizations that you can do to produce these plots in exactly the way that you would like to have them, this can of course be quite time consuming, and you don't want to have to read this every time you want to create a similar plot. We do have an option here where you can store these personal presets. So for instance here we will call this example plot ncam1. And then we know that this is the plot that we made when we were looking at ncam1 with all of its settings, and we can then store this as a preset. So now this is stored to our specific login, and we can then return to MegaSampler as we would have from the main page. And here under presets, you can see that we have example plot ncam1. We can simply select that, click next, and now all of the data sets have already been selected for us. So we no longer have to do anything here. If we would like to look at the exact same plot again, we can simply type in ncam1, or we can save all of the presets as we've had them before and look at a different gene. Simply click next, and we are then brought to the same plot that we had before with all of the settings already done for us. So now we've looked at an example of how you can look at a gene's expression across multiple selected datasets. However, perhaps you'd like to look at how this gene is expressed across a broader spectrum of datasets. And you'll notice that down below the graph there is something called View Expression in Many Datasets. So if we click on this, you see that we are then presented with all of the datasets that have been run on this same U133P2 Affymetrix array platform with the same data handling. And there you'll see that you have all the data sets represented in one graph, and you can hover over them to figure out which data sets are which will then perhaps allow you to select which data sets you would like to use in other MegaSampler analyses like we've currently gone through. So for instance, now we're looking at NCAM1, and you can see that if you hover over a lot of these high expressing ones, you see that they're all gliomas, medulloblastomas, neuroblastomas, and they're all neural-like tumors, whereas if you scroll over some of the data sets that are lower expression, you'll see that that you end up with blood cell tumors, for instance, or other tumor types that do not have as high expression of the neural marker NCAM1. So now we've looked at some examples of single gene expression across multiple data sets. If you are interested in being able to compare data sets and their gene expression, for instance, using differential expression across multiple data sets, please watch the video MegaSearcher instead, which will go into more detail looking at such analyses.